George, who are both leaders in their fields. Liz is Deputy Head of the Cancer Biology and Therapeutics Unit at the John Curtin School of Medical Research at ANU, and she's also head of the National Platelet Referral and Research Centre at ANU. Amy is leading the ANU Centre for Therapeutic Discovery at John Curtin. These incredible women are at the vanguard of cancer research in Australia and potentially the world. And they're here at CHARM today to update us on the amazing innovations in cancer research that are happening right here under our noses. So without further ado, I'd like to introduce Liz.
Thank you very much, Diane. Very kind, very generous words, and a pleasure to be here today. I'm actually filling in for Ross, who was here this morning but has got other meetings. But it's a... Do I have a point? Yeah. My job is to talk to you a little bit today about the Comprehensive Cancer Centre Research Collaborative, or the 3CRC, initiative that has been underway even before my time coming up to Canberra from Melbourne. And it's a venture that has got multiple partners, but primarily, underpinned by ACT Health interest, as well as the Australian National University, particularly the John Curtin School of Medical Research. And so the genesis of this initiative has come from several years ago, the establishment or the creation of a Centenary Chair in Cancer Research. And this was a joint initiative by ANU and ACT Health to celebrate the centenary of the ACT. And the goal of this chair was to enhance world-class cancer research at ANU and to facilitate collaboration between cancer researchers at ANU, principally John Curtin School of Medical Research, and the Canberra Region Cancer Centre based at the Canberra Hospital, clinicians and also other researchers based here at the hospital. And so this was formalised in April 2015 when Professor Ross Hannon was appointed, and a Department of Cancer Biology and Therapeutics was established at ANU. Now, this is the first department in Australia dedicated to cancer biology, which is, when I learnt that, I was actually surprised. And Ross Hannon was appointed to be head of the department. The other goal through this Centenary Chair appointment was to establish Australia's most advanced and academically led high throughput drug screening facility with the purpose of discovering new drugs as well as repurposing existing drugs for cancer therapy. I'm not going to talk too much about that today because Dr. Amy George is here and she can talk and in using much better frames of reference than I can. Also, the establishment of this screening facility was initiated because of a direct link with the National Cancer Institute in the USA. As part of the Moonshot initiative, we had access to a product discovery library. And so the department was created. Ross came up from the Peter MacCallum Cancer Institute and led it. It basically has built it together with his team. He brought up a group of maybe 6 or 7 individuals, solid, hardcore cancer researchers, and they built the department with this focus in mind, to focus on the fundamentals of cancer research, listing many of the strengths of the department here, together with translational models and access to models, so either existing models or the development of new models, which involve everything from flies through to rodents, where they could establish and test new reagents created and identified through the high throughput screening facility, but most importantly with this clinical arm where we collaborate and corroborate with clinicians that were already embedded at the Canberra Region Cancer Centre, with the view to developing and progressing reagents and drugs identified at the fundamental level and into a clinical space where they can be further evaluated and hopefully even progress beyond that. As part of the establishment, a set of recruits were attracted to Canberra, most notably Leonie Quinn's group. She's primarily, she works in Drosophila but also has mouse models and has a really keen interest in all mechanisms involved in cellular dysregulation of cellular growth, but with a keen interest in brain cancers. And Professor Barry Thompson, who was recruited from the Crick Institute as an EMBL Fellow. So through connections, we are now an EMBL site within Australia. There's EMBL sites all around Australia, but now at John Curtin we have an EMBL Fellow. And I came along for the ride. And so together, the group and building that department at an academically led basic research department was established. But in order to really complete that picture and in order to build and create what we now call a comprehensive cancer centre, and I'll talk to you a little bit about that definition, what we need is to integrate with the clinicians and clinician researchers, not only at ANU, not only at John Curtin, but also at the Canberra Hospital. And so this collaborative has been developed and is still in a work in progress, describing a partnership between ACT Health, ANU, University of Canberra, and we have just, in the last year, become partners with the Peter MacCallum Cancer Centre. And a memorandum of understanding was signed between these key players representing major arms of this collaborative centre in September last year. And the idea was to gain access to world's best practice, precision medicine, and essentially, leverage off the intel that the Peter MacCallum has had through being an established cancer centre for some considerable time, but then to build and become our own comprehensive cancer centre. So just a couple of other dot points I've got listed here. We are a founder, through ACT Health, a founder member of the cancer medicine program established at the Garvan, and this has enabled ACT patients direct access to treatments via the Garvan Institute for rare cancers. And Ross has really driven this philanthropic program to establish a comprehensive cancer centre right here at the Canberra Hospital, and with the view to invest in clinician scientists, genomic diagnostics, phase 1 trials, and clinical research laboratories. So I had to actually have a bit of a look. What is a comprehensive cancer centre? And so it's got its own wiki page. And Ross has designed this based off definitions that the NCI in the US has defined, where a comprehensive cancer centre must have expertise in laboratory, clinical, and behavioural and population-based research. And according to this definition, comprehensive cancer centres initiate and conduct early phase and innovative clinical trials and conduct other activities in outreach and education, as you can read here. So this is really the primary goal, to not just look at cancer in Canberra and to use the department and to use the existing facilities here through the Canberra Region Cancer Centre, but is to actually develop a comprehensive cancer centre. So whilst not making any judgment at all on the clinical treatment that's currently available, it is first class. This is a tertiary hospital, and it's the equal in terms of clinical management. It's a truth that Canberrans must travel in order to access certain treatments which are just not currently available in Canberra. There's also limited access to early phase clinical trials, limited access to bone marrow, allogenic bone marrow transplants, personalised cancer medicine, and the capacity to conduct translational cancer research doesn't yet fit that definition of what a comprehensive cancer centre has. So this is our goal, and this is what we've been working on, and I'm just the spokesperson today. This is really something that Ross has driven together with other people here at Canberra Hospital. So our focus has been upon attracting a clinician scientist with, clinical credibility as well as research credibility, who will be able to bring their group and in the process develop a clinical PhD training program. The laboratories, where we have the space at John Curtin, we also are looking to develop, or have a big part in the development of level 3 of the existing cancer centre over here on site. There are new services and clinical access to clinical trials, clinical cancer genomics. We're hoping very much to piggyback on the fantastic work that Matt Cooke and the molecular genomics facility have already built here, and they've made fantastic inroads, and we would only wish to join with them rather than replicate, because that access to high-level genomic analysis is really the way forward, and development of molecular pathology as well in concert with immunology. On the research side, I'll leave much of this, I'm going to let Amy talk about in terms of the high throughput screening facility, and then a list of all these other important points, some of which is going to be talked about later on today, I think, around palliative and end-of-life care research, to really flesh out a comprehensive cancer centre. And so this is just my final slide. This is the virtuous cycle of cancer research as described by Ross. And so just quickly walking you through it, we start with cancer research questions, and this requires much effort and skills and hard graft in the laboratory to push research questions through fundamental basic research and cancer models, piggyback or tap straight into platform technologies where we can begin to hopefully manipulate the cellular growth, and then through to, in what is, we hope, a seamless cycle into a clinical phase where we have cooperation both at ANU and here on site at the hospital, feeding into clinical trials and up eventually into what we're all looking for, new and improved treatments, and then the other KPIs which will come along for the ride. But of course this remains the major focus, of course, for clinicians, but also researchers very much care about where their research ends up. So on that note, I'll hand over to Amy to continue the story. Thanks.
Thanks, Liz. And I just wanted to, before I begin, thank the organisers for inviting me to present today. And as Liz mentioned, I'm going to be talking about some of the work that we're doing in high throughput screening space to look for specific drugs and biomarkers, particularly for cancer. And I wanted to talk about what we're doing currently and what we're going to be doing, hopefully, in the future. So just to give you a brief overview of what I'll talk about today. So I'll give you an introduction about high throughput screening and the types of equipment that we use to perform screens. I'll then briefly talk about the ANU Centre for Therapeutic Discovery and the equipment that we have and the services that we can provide to researchers here in Canberra. I'll then talk about an example screen that I've been working on, Not only do I manage the ACTD, I also have a research position within Ross Hannon's lab at JCSMR. And so I'll talk about that. And then finally, I'd like to talk about the future of drug discovery and sort of tap into the 3D screening and personalised medicine space and how we're looking to expand our screening capabilities here in Canberra. So high throughput screening is a really important method that we use in drug and biomarker discovery, and so really what it enables us to do is to move from testing on the lab bench in petri dishes, or this here is a 6-well multiplate, expanding that into something where we can test multiple targets. So we can work in 96, 384 and 15 36-well plates, which have an approximate size of about this. So it's quite small. And so for high throughput screening, what we require are robotics and automation platforms. So they're sort of key to being able to have higher throughput for drug discovery. We also require biological and chemical compound libraries, which could be either large scale, so they can be in the vicinity of 500,000 compounds, right down to some boutique library screens, so 100 compounds. We require designated assay readouts, so these could be plate reader-based assays, imaging-based or phenotypic readouts, or flow cytometry, and I'll touch on these in a minute. And then finally, we have a lot of data that come out of these screens, so we actually require some streamlined data analysis approaches and bioinformatic pipelines to make sense of the data that we acquire. So the types of equipment that we'd have in a typical high throughput screening facility include instruments to measure assay readouts. So here we have a high throughput plate reader and a high throughput microscope. We have liquid handling robotics, so we have precision liquid handling robotics that can go down to nanoliter dispensing, right through to bulk liquid dispensing robots. We have specialised microplate incubators and storage towers, which help to facilitate the growth, maintaining the cells in these at these conditions, because you have smaller volumes, so you do require specialised storage. And then all of this is kept or conducted by automation. So we have robotic arms that feed plates in between different pieces of equipment as and when required. And so we primarily work a lot with cell-based models and cell screens. So just to give you an overview of how we'd actually do one of these screens. So we take one of our microplates, whatever size that we were screening in. We would then put the cells into these microplates. Usually we do this using bulk liquid dispensing robotics. And so this allows us to have individual cells in each one of these wells in a plate. So then at that point we choose the reagent that we wanted to screen with. For example, we might actually want to knock down or knock out a specific gene of interest. So we could use either RNA interference or CRISPR technologies to do this. We can also use compounds, we can screen viruses, all sorts of different things. And we usually deliver these into the plates using the precision liquid handling robotics. At this point, we'd have a specified time period where we would need to conduct some sort of time point or end point of the assay. This can vary amongst all sorts of different types of screens. But essentially there's 3 major ways that we do that within the ACTD. So we have a high throughput plate reader where we could look, for example, at cell viability. So if we're trying to discern whether or not specific drugs are killing cancer cells, we might want to look for reduction in viability. We might be certain biochemical reactions of interest. We have a high content imager, which is essentially a microscope in a box. We can do all sorts of really cool things with this instrument. So we can look at cell shape, size, morphology. We can look at subcellular localization of specific proteins and staining intensity, and I'll give you an example of that later on. And also we've acquired a high throughput FACS analyzer. So we can do very similar things. We can look at cell number, size, morphology as well as staining intensity. And so these 2 instruments really allow us to perform phenotypic screening. So I just wanted to touch briefly on phenotypic screening. This is one of the topics close to my heart. I really enjoy this part of my job is looking at lots of images. And so what higher content screening allows us to do is quantify biology from images generated using automated cell imaging. So what really this involves is that we take our cells and we stain them. We can use multiple or single fluorescent dyes to do this. We then take these microplates and we image them on this high throughput microscope that can capture many images of the cells. And then we can take these images and we can extract information out of them. So, for example, and these are very sophisticated software packages, so we can do all sorts of really exciting things. But just to give you sort of an overview is that, you can take images that look like this. For example, nucleoli, we're interested in the number of spots that's in this particular nucleus, for example. So you can actually input this into software that allows us to look at individual cells, look for the phenotype that we're specifically interested in, and then be able to train the software to recognise that specific biology and perform and enable a quantitative readout. So now I just wanted to briefly switch to talk about the ANU Centre for Therapeutic Discovery, or ACTD. As Liz mentioned, we're located at the John Curtin School. We're essentially in the front door, so as soon as you walk into the building, you'll see us. We're a fully automated high-throughput screening lab where we screen mouse, human and other cell lines and cell-free assays. Our facility was funded by the ACRF, the ANU and ACT Health, and it opened back in 2017. Our equipment costs is approximately $4 million, and I guess the important thing to note is that we're open not only to users at ANU, but also any other researchers that are interested in both in the Canberra region and also Australia-wide. We've even had clients from New Zealand as well. So if anyone's interested, please head to our website and we have more information. So just wanted to sort of give you some context about the types of robotics that we have. I just wanted to show you a short video of our robots in motion. This is one of our screening workstations. And essentially, we're just doing some drug treatments in this particular video. And this here is our robotic arm. So this services all those different pieces of equipment that are in this particular cabinet. At the back here, we have our precision liquid handling robot that's treating a plate of cancer cells with drug. And it's just moving the plate into a position which allows us the robotic arm to come in and collect that plate. Because we work mostly aseptically, we need to be able to protect the cells. The cells have, there is a lid that will be put back onto this plate. We have equipment that essentially is just suction cups here that hold the lid for us and drop it onto the plate when we need it. And then after it's done this, it can move it to the next piece of equipment that's required. So in this case, it's the high throughput plate reader, which is up the back here. I have more videos. They're on YouTube. If anyone's interested, please have a look. So in terms of the services that we offer, we're very much a user-assisted screening facility. So sort of leading on from what Liz mentioned, we're very academic, very collaborative. We like to work with researchers. We think the best outcomes come from working with people that understand and know their specific biology. We consult with people about screen design and assay workflows. I provide assistance with grant applications relating to screening aims. So if anyone wants to get the words right in their grant around it relating to a screen, or if they want a budget, they can come and talk to me about that. We enable access to biological and chemical libraries for screening. We do on-cell screening consumables and reagents. But I guess the other thing that I wanted to mention was that we do work a lot with researchers on boutique applications and pilot studies. So if there's anything that you want to try out and see if it works, we're more than accommodating for that. So now I just wanted to touch on an example of a screen that I've performed. So when I wear my research hat, I'm interested in the nucleolus in cells. And the nucleolus is a dynamic non-membrane-bound organelle which sits within the nucleus of cells. And its primary role is for ribosome biogenesis, or the generation of protein factories in the cell. And here's just a picture which I actually took on the high-throughput microscope showing individual components of the nuclei, and when we merge them together you can see the core and the periphery of these organelles. But one of the key interests that I have regarding the nucleolus is that it has an ability also to sense cellular stress and make decisions about cellular fate. And this is a process that we call Nucleolus Surveillance. And in this process, what happens is if a cell detects stress, it increases the stabilisation of p53, which is a guardian of the genome. And this leads to cell cycle arrest, apoptosis and senescence. And so the nucleolus can sense very specific types of stress. This includes DNA damage or genotoxic stress, inhibition of RNA polymerase 1, which is required for ribosomal RNA synthesis, and that constitutes the ribosomes. Oncogenic stress, so overexpression of oncogenes like MYC and RAS, and also mutations or loss of ribosomal proteins, which are observed in a group of diseases called ribosomopathies, which is also another one of my specific research interests. So what we wanted to do in this case is that we wanted to screen FDA-approved drugs to identify modulators of this response. And we wanted to do this for two reasons. We wanted to look for drugs which we could repurpose for the treatment of ribosomopathies. So in ribosomopathies, p53 expression and stabilisation is aberrantly activated. We wanted to look to see if we could find drugs which modulate and reduce that p53 response. Then also we're interested on the flip side at understanding which specific molecules further enhance the nucleolus stress response, so further enhance p53 expression. And so I'll talk briefly about CX5461, which is a polymerase 1 inhibitor, and our work in the cancer therapy space. And so, using FDA-approved molecules may be an option for us to develop and fast-track potential therapies for diseases where this process is important. And so to do this, we generally have cell models for screening. So in this particular case, we took a human lung adenocarcinoma cell line, which is wild type for p53, and we depleted these cells of RPS19. And so what this actually does is it enhances the expression of p53 protein and downstream of that p21. And when we look at these cells underneath a microscope using immunofluorescence staining, when we deplete RPS19, we see an enrichment or an increase in p53 protein intensity, which is localised to the nucleus. So based on this particular phenotype that we see under the microscope, we can actually utilise this for scoring our screen. And so what we did was we took this model, and then we treated with 4,169 different compounds, which were either FDA-approved drugs, kinase inhibitors, or epigenetic modifiers at 4 different doses. And then after 48 hours, what we did was we fixed, using robotics, we fixed and stained these plates to look for the nuclei. We do that with DAPI staining. We do this to count the cell number and p53 protein expression. And so we were interested in looking specifically at nuclear-expressed p53 protein. And so from our screening campaign, this is just a summary of the entire result. So we were interested in the cell number and the p53 intensity. As I mentioned, I do have a specific interest in ribosomopathies, and so we're interested in those candidates which reduced p53 here, but had no negative impact on cell number. And so some of these may be potential ribosomopathy therapeutics, and we're actually testing a number of these therapies in preclinical models of a ribosomopathy called Diamond-Blackfan anemia, which is currently ongoing, but showing some promise. I just thought I'd talk today about some potential cancer therapeutics that came out of this screen. So when we induce nucleolus stress and then we treat with these compounds, we see a further enhancement of p53 and a further reduction in cell number. So we're very much interested in looking at these particular compounds, in particular a compound called compound 20 or C20, and potentially using this in combination with other drugs that induce nucleolus stress. And so as I mentioned previously, CX5461, which is a polymerase 1 inhibitor, in the Hannon lab we've shown that this particular drug does induce nucleolus stress in a B-cell lymphoma model. It does enhance the specific activation of p53, as you can see here, upon treatment. What was most exciting, I think, is that as a single agent, it significantly increased the survival benefit of this very aggressive lymphoma model. And so our rationale here was that treating the drugs that we identified in our screen with and using those in combination with CX5461 may further enhance the therapeutic benefit for the treatment of cancer. So in our lab, we're specifically interested mostly in hematological malignancies, and in this case, we had a model of acute myeloid leukemia, or AML. AML is a cancer of the blood and bone marrow, and it largely affects older adults and elderly. but unfortunately there are fairly limited therapeutic strategies for these patients, particularly for the more aggressive forms of the disease. And so in our lab we have a specific model which has an MLL-ENL translocation, which is quite aggressive. This model also expresses oncogenic RAS and is wild type for P53, and it's been published by Johannes Zuber's lab back in 2009. And so what we do with this AML model is we take these AML cells, and these cells have GFP and luciferase tagging. We transplant them into recipient mice, and then we allow these to engraft. After 7 days, we perform bioluminescence imaging. And then on day 8, we start to treat the mice either with single agent CX5461 or C20, or in combination. And then we monitor the mice using weekly bioluminescence imaging and Coleman ethical endpoint. And so on the right here is some data with the bioluminescence imaging. So essentially, just to sort of, in a nutshell, the more colourful the mice are, the higher the tumour burden. And what we noticed was when we treated C20 in combination with CX5461, we increased the survival of the mice with AML. So as you can see here with this Kaplan-Meier curve, when we treat with single agent, both single agent, we do have a small survival benefit. But when we treat in combination, we see much enhanced efficacy. I guess what's most exciting is that this drug is quite an old drug. We've moved to a second generation version of this drug. We see the same effect. So at the moment, we're working to try and figure out the mechanism of this combination, and we hopefully will publish this soon. So in the last sort of 10 minutes, I wanted to talk about the future drug discovery efforts that we're undertaking. So I guess traditionally large-scale cell-based screens have been performed in 2D cell culture models. And as I've shown you, that's been quite good in our instance. We've actually been able to identify some therapies that are working in preclinical models. However, 2D cell culturing may not will accurately reflect the complexities of a 3D environment. And may have implications in the identification of relevant therapies for disease. And I just wanted to sort of make a point here that, in terms of pancreatic cancer, for example, there's been limited drugs that have been identified for the treatment of pancreatic cancer in the last 20 years. And I guess some of the work, the main clinical drug that's used to treat pancreatic cancer is gemcitabine. And this was work performed by Vicky Avery's group, who are located at GRID in Queensland. And what they show, they take these pancreatic cancer cell lines, and they culture them either in 2 dimensions or 3 dimensions as spheroids. And so what you can see is that when you culture these particular cells either in 2D or 3D, that there is a difference in the level of inhibition. And when you look at these cell lines specifically, you see that there is a reduction in efficacy of these drugs in 3D. So it is suggesting to us that perhaps we do need to rethink our drug discovery efforts and start to think about more relevant models when we do our screening. So I guess for a long time, some of these things that we culture and reagents that we culture these cells in are really, really expensive. And so it's actually very cost prohibitive as academics to perform screens using 3 dimensions. But I guess there's been, at least in the last 5 years, really a lot of evolving technology that's enabling us to actually start to think that we can actually do this in high throughput. So this is advances in 3D biology, microfabrication, so producing hydrogels that can propagate all the sorts of different cells and different models that we're interested in, as well as tissue engineering techniques. And so I guess what's really exciting as a screener is that we're now able to readily perform high throughput screens using spheroids, organoids, co-culturing, organ on a chip, and 3D bioprinting, and I'll talk a little bit about 3D bioprinting. And so just wanted to show you here that there's a company in the Netherlands called Mimetis who are developing organ on a chip technology, and this here is a microplate which has 96 individual chambers where you can actually grow mini organs. Which is very exciting for drug discovery. And I think Roche are currently using that in some of their screening campaigns. But I guess also what's really exciting is now we can start, instead of just using cell lines, we can start thinking about culturing primary cells, iPSCs or other complex models, and really start to drill down on the genotype to phenotype biology. And it'll also allow us to, open up possibilities for us to take patient samples and perform personalised medicine, so be able to identify specific drugs that are going to treat a certain person's cancer better than others. And so what our future vision at the ACTD is to expand our current capabilities and perform 3D and complex organism screening. So to go from the two-dimensional mouse and human cell work that we do to 3D human and cell work, as well as incorporate a number of small organisms as well in our screening campaigns. To facilitate this, two of the most important technologies that I'll discuss today are 3D bioprinting, but also the custom-built light sheet microscope that we have plans to build, and we're trying to get funds for this at the moment. This will be done in collaboration with Dr. Steve Lee, who holds a joint position between the John Curtin School as well as the ANU College of Engineering and Computer Science. And that will be hopefully the second, only one of two purpose-built light sheet microscopes in the world. So as I mentioned, 3D bioprinting will facilitate a lot of the development in our facility, we hope. And what we're doing at the moment is partnering with Inventia Life Science, who are a company in Sydney that work on 3D bioprinting. There are a number of different companies that offer bioprinting solutions, but we want to go from just sort of a box where it's quite limited to being actually able to do multiple plates at one time. And so this is just a video. I just wanted to show you a video from Inventia. This is their standalone instrument, but the technology will be very similar. And essentially what we have here is you're able to have your specific hydrogels of interest. You're able to put up to 8 different cell types into these tubes for printing. Then you'll have your waste and your sterilisation. And as soon as that goes back in, you can essentially start printing straight away. It's all computer operated. And this is just one way to do this. This is spheroid formation. So you can build a scaffold, a hydrogel scaffold for your spheroids, which is quite nice. It's very reproducible. And within this well here, you can, or in this groove here, you can actually print your cells, and they can actually form spheroids within, they say here, 48 hours. I guess it's cell type dependent. But I guess what this really enables us to do is to start thinking more laterally, and so we can do all sorts of different things, like here they say lymphocyte infiltration, as well as migration. But there are all sorts of other different things that this will enable us to do. So apart from spheroids, we can look at spheroid invasion, migration towards one chemoattractant or multiple chemoattractants. We can co-culture spheroids with immune cells to better model some types of cancer, for example. We can grow organoids. We can do chemotaxis with a chemotactic gradient, as well as other organotypic models. So it's really quite exciting as a screener. And I just wanted to touch briefly on some of the work that Inventia have done with spheroids. So they've provided me with this picture to show you that, so here we have a spheroid which is stained with DAPI, which is in blue, and green is Ki67, which is a proliferation marker. And this was taken using light sheet microscopy, so hopefully similar to what we'll eventually build. And I guess the key point here is that when you look at both of these, it really doesn't look like there's that much difference. And that's what's really exciting about this, is that instead of us having to, as scientists, manually sit there and seed these cells into plates, we can actually get a printer to do it, and to do it very effectively. So that will enable us to perform drug screens, or higher throughput drug screens. I just wanted to finish and talk about one of the projects that we're quite excited about once we have the bioprinting facility. This will be in collaboration with Leonie Quinn and Guillermo Gomez, who's at the Centre for Cancer Biology in Adelaide. And they work specifically on brain cancer and glioma. And the treatment options for patients with glioma is fairly limited. And there really have been no therapies identified for around 30 years. And so what they hypothesise is that printing 3D mini brains might be able to better model brain cancer and allow us to identify more relevant therapeutics for brain cancer. And so this is just a schematic of how they plan to approach this. So they plan to take human iPSCs, they either genetically modify these or not, depending on type of assay that they want to do. Then they'll seed these into these microplates that will form brain organoids. We'll do this using the 3D bioprinter. And this here is just an image that was taken. This was taken using a high throughput confocal microscope, where you can look and see that these structures form what we call mini brains, and you can stain and get so much information out of these specific structures. So that will hopefully aid towards identifying new therapies to treat brain cancer. So just in summary, I just wanted to say that high throughput screening is a really efficient way for us to test many agents or conditions on a large scale. We can use this to identify not only novel or repurposed drugs, but you can perform combination therapies to treat diseases such as cancer. The newer screening technologies are really, really exciting, and it allows us to better model disease and to identify more relevant therapeutics more quickly. And our vision really is to develop a complex organism screening unit within the current ACTD, and that hopefully this facility would place us at the forefront of this technology in Australia, and also provide the Canberra region with a really cutting edge capability. And finally, I just wanted to plug our upcoming Functional High Throughput Technology Screening meeting, which is our 10th annual meeting, and it's happening at the John Curtin School in late October. And our program will cover high throughput screening, 3D screening and models, personalised drug discovery, so everything that I've talked about today. And we're just about to go live with our registration, so for anyone that's interested, please head to our website for more information. And finally, I just wanted to thank, there's a lot of people involved in these long screening campaigns, so there's many people to thank, in particular Ross Hannon and the Hannon Group at ANU for all of their assistance. And with that, thank you, and happy to take questions.
You've got it there. Well, thank you both very much. What an exciting opportunity for Canberra, really. We are the nation's capital, but we are a small town too. So, and I think anybody who's struggling with a loved one with cancer will know how frustrating it can be, actually, for clinical care. Has anybody got any questions that they'd like to ask? No problem, because I have got some questions. Oh, great, okay. They're suitable? Okay, all right. Okay. Well, can I take those then from you, Hannah?
Yes, I think that's supposed to be.
It happens to me all the time. Well, I'm going to start off by asking you, how long do you think it'll be before there's a palpable difference felt by the Canberra community from this amazing initiative?
Well, already I'm not the best placed person. I know Dr. Crispin's up there, who probably could even answer how many patients has he sent to the Garvan Institute to take advantage of the MOST trial, MOST trial, where his patient can now have genomic analysis of their tumour, and then it is the sequence is screened against libraries and new candidate molecules which either are in trial or about to go into phase 1 trial. It throws up potential candidate treatments which are not yet FDA approved or TGA approved, but in theory could be accessed by this patient. So I know of some individuals where that's already happening. Oh, sorry. Do I have to repeat all that again?
No, I don't think so. Did everybody hear that?
Yeah.
So there may be changes. We may be feeling changes quite quickly. Dr. Grissom, did you want to comment on that?
Number? Sorry to throw you in here, Phil.
I think there are a variety of mechanisms where we can access more personalised approaches to cancer management. I think the reality is that for some of this, it's an opportunity. It's an opportunity for lots of people, but the numbers of people who benefit are still relatively small because of the number of compounds that are available. And so actually, when you look at these two initiatives together, the high throughput screening, where potentially we can access more and more compounds, even things that generate ideas, things that are readily available, the opportunities here could expand enormously. So whilst it's affecting a lot of people in a small way now, and a small number of people in a large way, I think the opportunity is enormous in the future.
Thank you.
Yeah, I think we are the nation's capital. Yes, we're a small territory, but we're the nation's capital, and that's where, we have advantages over Sydney, Melbourne, and, I referred a couple of times to the National Cancer Institute in the USA. Well, they're based in Washington, and they coordinate comprehensive cancer centres all over the US. Europe have the same system. So we have comprehensive cancer centres in Australia, but there's no network. There's no, linkage. They're kind of isolated islands, and I'm sure a lot of competition. I don't work directly in that space, but I can imagine. But here we have the opportunity in Canberra, because we're the capital, that we can actually, at least take a leading role in coordinating all the sites, and helping to drive that academic clinical partnership, which, is so rare. That's why that MOU was signed with Peter Mac, because they're probably one of the only examples of a real academic research institute looking at cancer, sitting right next door to a leading cancer treatment facility. And so we have that same opportunity here. That's basically what we're pressing, and that's through the ANU site as well as here in Building 15, where we would embed clinician researchers and researchers together, along with all those other umbrella, requirements that are also necessary, such as patient, well-being, patient health, community outreach, all of those other key components.
Yeah. And does Greg Hunt? Oh, sorry. Yes.
Carrying on from that is the fact that we're also able to collaborate. The ACT is being asked to collaborate in national initiatives. So the MOST trial, we were the first to come on board with New South Wales, and that then led to the Australian Cancer Genomics program, medical program, and a centre with $50 million injection of funds, which we're part of that. We're one of the foundation members. So not only can we lead, but we're also part of national collaborations which lead to these types of discoveries working together.
So I'm going to ask a question that's come from online, and somebody wants to know how much all this costs, because clearly it costs an awful lot. And is it funded through just grants, or have you got a lot of government funding?
So, Amy, you talk on your facility first.
So at the moment, the current facility that we have is being funded by the ACRF, which is Australian Cancer Research Foundation. So that was probably roughly half of our current facility, as well as ANU and ACT Health. So getting that funding from philanthropy certainly helped us to leverage more money from ANU and ACT Health. But yeah, it does cost a lot of money. It costs a lot of money to not only operate the robotics, but to keep maintaining the robotics and keep on top of all of the technologies that are required to be able to do cutting edge screening. So yeah, it is expensive. It's in the millions of dollars. And to go through this initiative that we were talking about, it would be in the millions of dollars that we're looking at.
And so just a couple of things. So, we buy nearly everything from overseas, right? You just think about how much is imported. So we have to pay import and shipping costs as well, and that builds up. In terms of the vision that we were talking about earlier, to recruit outstanding individuals, yes, that takes a lot of money because it's not just the one person, they come with their group. But also there's people here, and I know Ross has really advocated buying back some of the clinical time we've got some fantastic clinicians here who have really brilliant research minds as well, but they're absolutely swamped with clinic. So the idea would be to buy back some of that time to free them up to work 1 day a week, taking an explanted culture from a patient and growing it in an organoid culture and then screening it against 4,000 compounds, just as almost a blue sky experiment. But as Phil was saying, that's the opportunity exists.
Yeah. So making these three-dimensional sort of models, if you like, does that mean you don't have to use mice anymore?
Well, that would be the plan.
Yeah.
I think mice have their place in medical research, but I think at least now we can take patient cells, so cells directly from patients, and screen those. That's what's I guess the most exciting thing. We can kind of do that currently, but not. If we take cells out of particular patients, sometimes we can't actually grow them on plastic. So having these systems where we can actually grow them in where they're going to be happier means that we can actually facilitate this a lot better.
Yeah, fantastic.
And there's huge evidence that 3D organisation is absolutely critical. So Amy showed a little bit of data, just how a drug affected the cell grown in 2D versus 3D. But I'm sitting on a panel right now reviewing grants, and they're all cancer grants, which is probably not my best expertise. But the number that are looking at 3D niche biology, so building bone marrow niches, that like a 3D matrix where you have so you've got stromal cells, you've got invading macrophages, you've got other cells, my favourite cell, megakaryocytes and platelets, building that niche is clearly the way Australian research is going and world research is going.
Yeah.
And so this type of approach is really getting ahead of the pack, because I'm pretty sure no other screening facility in Australia would be offering this, or they're probably all thinking.
Minor.
But
We're going much larger scale.
Yeah.
And how you're planning to do it actually? You're taking the cells from the patient, but how you're planning to grow inside to make a 3D culture?
I think the gel has media.
Hydrogels. So depending on the cell type, there's all sorts of different hydrogels that you can use. So we would take those cells and put them into the hydrogel that's most appropriate, and then we would print those into the plate using the bioprinter. Yes.
So the question was around the growth of the cells in 3D. Yeah, it's like growing in RPMI versus DMEM. So you need to figure out, cells will grow in lots of media, but what's their optimal one?
Yeah. Absolutely.
Yeah, so there will be a lot of optimization to grow the cells in there.
Sure, just like in 2D.
I'll just quickly go on from that. So working with Inventia, they're working a lot with researchers, and they have a lot of different options available for certain types of models already in place. So they've already done part of the optimization. Of course, once we do get the instrument, we'll need to do optimization, and optimization will be different for every cell type, but at least if we've got an idea about where to start, it's going to make up the process a lot easier.
So I've just got one more question from online, and that is, are all these plastics recyclable?
Thanks, Liz. Unfortunately not. I think as a scientist, when I see the amount of plastic that I go through, we're all conscious of what we go through. Unfortunately, that's part of it. But we do. Well, most of us, I can at least say myself, I am conscious of what I use and try to reuse what I can when I can. So.
But you've got 1,500 wells on something that's the size of your hand. So
So we do reduce the volumes of things. Sorry, we do reduce the volumes of things that we use. So if we were actually doing this all by hand, we'd probably use a lot more plastic. So actually miniaturising things the way that we do reduce that. So.
Well, thank you both very much for coping with all of those questions. And I'd like you all to put your hands together. And thank you very much. It's now afternoon tea time, slightly extended, and I'd love to see you all back at 3:10 to listen to how our Health in Our Hands strategy initiative is going, and I think that'll be Jane Ezra. So I hope you'll be back to listen to it with as much excitement as I will. Thank you.
Thank you.
That was such an exciting initiative.
I know, we're excited.
Everything that goes pinned.
I know, we just need some money.
Yeah, I know. The other question I was going to ask, but I thought that's boring, is what can the community do for you? You know, this is an amazing thing, so how can the community help you?
We're always open to philanthropy,
Yeah.
so we do speak a lot with potential donors.
Yeah.
So and we invite donors to come and talk to us, come to see the facility, come to see what we do.
Yeah, I think it's
Cancer space in Canberra, and that's it. It really helps facilitate. They see what we do and bring it.
And the other question I was going to say, does Greg Hunt know all about this?
Okay, good afternoon everyone and welcome back to the first day of CHARM. My name's Liz Gardner from John Curtin School of Medical Research at ANU. It's my pleasure to chair this session of stellar speakers talking on different topics, even though some of the names of the titles are within range of each other. It's my pleasure to first introduce Professor Matt Cooke, who's the Director of Immunology here at the Canberra Hospital. He's also the Director of the Centre for Personalised Immunology at John Curtin School of Medical Research, ANU, as well. So he wears two hats. I think today he's going to talk on the ANU Grand Challenge, in which he was a leader in that successful bid, and give us an update on progress made in that endeavour. Thanks, Matt.
Thanks very much, Liz. And thanks to the organisers for the invitation to myself and Jane Desborough, who's going to share this session with me to discuss this Grand Challenge, the inaugural ANU Grand Challenge. Just so that everyone's aware, this is a program of substantial funding which the ANU announced to support interdisciplinary projects, and that's certainly what ours is. I'm going to focus on the aspect of the project that I'm leading the charge on, and Jane will do the same, but along the way we'll mention some of the interdisciplinarity. And overall, our grand challenge is to do with the personalisation of medicine, what we call deep personalisation of medicine. And what we're seeking to do is to assist in the transition from our approach to medicine, which is summarised in this slide, and which really remains the paradigm of clinical medicine, where the phenotype dominates. As indicated here, clinical assessment, investigations and training of medical students and recruitment into clinical trials is largely determined by the phenotype, but of course the phenotype is the outcome of genotype environment interactions. So put another way, variation in the phenotype is the result of variation in the environmental exposure and genotype. This is a simplification. It's not always a simple summation, but these are the variables. So phenotypic variation is performed in this way, and in the clinic, clinicians are expert at phenotypic variation, and this is where we've concentrated. A lot of effort has gone into characterising environmental variants. These are sometimes referred to as risk factors, so things like diet and occupational exposures and other activities. But of course, medication is a subset of environmental exposure as well. But what we haven't done too much is take into account genomic variation, and this is because it's been largely inaccessible for all but a small number of rare diseases up until recently. But the advent of genomics means that this is potentially changing, and therefore we can start to move away from this exclusive attention that's paid to phenotypic variation. Now, why is this important? Well, this is just an example of high-profile study published in the New England Journal of Medicine, looking at patients with septic shock, looking at various forms of intervention, but particularly the use of corticosteroids. So effectively, an intervention which is occurring right at the far end of the disease process. And this is a negative study. And so what we're seeking to do by moving away from attention to the phenotype is to move more proximally in the disease process where the pathophysiological events occur, so that we might move away from these sorts of negative results. Okay, so let me just define these three terms. Conventional medicine, where we might think about treatments which are directed at patients according to their phenotype. And so an example might be the use of aspirin, a very widely used drug, and candidates for this therapy are arrived at according to conventional diagnostic approaches. Precision medicine is where we use a treatment which works on probably a single molecule in the body. So we know in detail how this medicine works, but the diagnosis might still be phenotypic, but the medicine is being used in a very targeted manner. Personalised medicine is where we have a precision therapy, but it's being targeted to individuals according to a precise understanding of the mechanism of their disease. And at the moment, the best way we can arrive at that understanding of disease is through genomics. And so a targeted therapy for cystic fibrosis is an example of such a personalised medicine. Now, is this important? Well, here is the results just of a detailed survey of the sorts of outcomes observed in studies published in the New England Journal of Medicine across this interval from 2003 to 2018. This was done by Nikita Parkash, who was a summer student here last summer. And what this shows very simply is the outcomes from these three sorts of interventions. The filled bars indicate the proportion of studies that met their primary outcome. And so you can see, first of all, that they're sort of on diminishing returns for conventional treatment, where the proportion of studies which are meeting their primary outcomes is falling. By contrast, for precision and especially for personalised therapy, primary outcomes are being met at a much higher rate. Now, this is important because here's a list of the most expensive drugs in Australia just in the past year, and you can see that this list is very much enriched with precision medicine, so drugs that work on a single molecule in the body. So they're all shown in red, but only one of these is used in a personalised way, where we target the drug to the individuals who are most likely to benefit from it, even though it's a precision medicine. And this is only possible once we understand the mechanism of disease with sufficient accuracy, and when we have the biomarkers that enable us to identify those individuals in the clinic. And until we achieve that, then there's a risk, a twofold risk. The first is that we might not be able to demonstrate the efficacy of what is a highly effective treatment if it's not given to the right subset of individuals. And secondly, that individuals who aren't going to benefit are still placed at risk of any toxicity from that treatment. All right, so the Ohio, our Health in Our Hands Grand Challenge is aiming at deep personalisation of treatment. I'm going to talk about genomics and bioinformatics a little bit more. But we have another program which is run by Antonio Tricoli, which is to look at the use of wearable devices to enable ongoing monitoring of phenotypes. Another program which is attempting to integrate data from both sources. And then cutting across all of these activities is the patient experience, which is what Jane is going to discuss in a minute. And we're testing all of this in using two cohorts of patients, first with multiple sclerosis and the other with type 1 diabetes. So if we come back to this scheme, then we're attempting to personalise medicine by starting to characterise genomic variation and then working out ways in which this information can be integrated into clinical practice. To understand how precision therapies might actually be analysed and how information from those therapies can be captured more efficiently. And then still looking at the phenotype, but again in a renewed manner by monitoring patients so that the phenotypes are collected not just when the patients come to the clinic, but continuously. And as I said, this is going to be tested in type 1 diabetes and multiple sclerosis. Now, I'm going to talk about some data that we have that give us some clues about how we might approach this problem, and what I'm going to tell you about has been performed by members of my group here, and also in collaboration with my colleagues at the Centre for Personalised Immunology, and also some colleagues down at Monash. So if we think about how genomics is giving us an avenue into personalised medicine, then really this has all happened in the 21st century. The human genome sequence became available in 2001, and then things really moved ahead because of technical advances which transformed the way we can actually perform human sequencing. And then we sequenced enough individuals around the globe to characterise the extent of genomic variation, and these three things enabled the implementation of whole exome or whole genome sequencing so that it now becomes part of clinical practice. And so for single gene disorders or Mendelian disease, this has already proven to be transformative. So if we take almost any phenotype where we suspect that there's a high incidence of Mendelian disease, we can solve these cases in 20 to 40% of presentations. But really the challenge is how we start to bring genomics to bear on complex disease. So let's think about these two conditions that we're focusing on as model complex diseases in Ohio. First is type 1 diabetes, and the second is MS. They're both thought to be immune-mediated diseases. Type 1 diabetes typically early onset and has a very strong MHC association. MS is different, poses different problems, heterogeneous, and there are already lots of precision therapies which are in place. Now we know that there's a significant genetic contribution to autoimmune diseases, and that's solid evidence for that comes from looking at monozygotic twin concordance, and that's what's shown here, so that you can see that the concordance rate for type 1 diabetes is pretty high. Now if we think about understanding how single gene disorders might be contributing to immune-mediated disease, here's some data from Genetic Immunology Rare Disease Flagship. This is a flagship of the Australian Genome Health Alliance, and this is seeking to characterise the frequency of single gene disorders that can be diagnosed through whole exome or whole genome sequencing. And in this study, we're enrolling people with different phenotypes, but you can see within the autoimmune category, we're still diagnosing about 25% of cases by whole exome sequencing. It's perhaps a bit of a surprise that the diagnostic rate is as high as that. Remember, this is selected, these are early onset or childhood and adolescent onset disease cases. So progress has been made applying genomics to monogenic disease. How do we make progress and extend this into complex disease? Well, as the previous slide illustrates, rare variants can cause complex disease, and I'll show you a couple of more examples of that in a moment. So this is one simple way in which we can start to apply genomics to complex disease. A single variant might turn out to be causative. But we can also learn lessons from elucidating diseases, monogenic diseases, elucidating the mechanisms in those monogenic diseases that we can then use to inform us about mechanisms of more complex cases. And then we can also draw on information from complex diseases that are treated with precision therapies, essentially phenocopying monogenic disease. All right, so let me just give you a couple of examples of how this might work. So here is a kindred that we started investigating a few years ago. A single individual proband with complex phenotype combining inflammatory and immune deficiency disease. and then it came to our attention that there was another family with similar manifestations. This is a family from Canberra. This one is from Tokyo. And more detailed analysis revealed that both kindreds shared abnormalities in their circulating lymphocytes, so T cell deficiency, subtle B cell deficiency, and hypogammaglobulinemia. So we sequenced this proband. Whole exome sequencing was performed, and we identified a de novo variant. So that means it wasn't present in either of her parents, had arisen somewhere in the fertilisation process. And this is a single missense mutation encoding an amino acid change in this protein, IKK2, encoded by IKBKB. And the work that I'm showing you has been performed by Chalisa Cardenas and Baha Miragazadeh and Liz De Silva in my group, and our collaborators in Tokyo. Right, so we had two families. We'd identified a novel mutation, but couldn't really be sure that we had the causal mutation because we couldn't be sure based on a statistical analysis given such a small number of individuals. But the mutation affects a residue that's highly conserved in evolution. And so in collaboration with Gaetan Bergio, we were able to generate a mouse that carried precisely the same mutation, the orthologous mutation. And then when Chalisa analysed the mouse, we found that the changes that we'd observed in the patients were recapitulated in the mouse model. So now we can be much more confident that this mutation is causal. And so this represents a new syndrome, and the point I'm illustrating here is that we can go from very rare individuals. We've got a technique for proving causation even when individuals, there are very few individuals. And once we can start to characterise these mutations, it gives us insight into mechanisms. Let me show you a second example. Here again, we've got a single kindred, an interesting phenotype combining features of autoimmune disease, so organ-specific autoimmunity, alopecia, inflammation of the pituitary gland, and thrombocytopenia, combined with antibody deficiency. And again, through whole exome sequencing, we identified a novel mutation and characterised that as the causal mutation biochemically. And now more recently, we've sought to understand the mechanism of autoimmunity in these individuals, and again have done this by generating mouse models using CRISPR-Cas9 to recapitulate the defect that is present in the patients, but this enables us to do analysis that we can't do in the patients. And so this has been done by Rashika Wirasena in Steve Daly's lab and Ainsley Davies in my lab. And so the point is that we've got a complex pattern of organ-specific autoimmunity. And so how do we work out the mechanism? Well, because we have a mouse model, we can start to dissect the mechanisms. And here the methodology is crucial. We've been able to identify the antigen-specific T cells using a particular reagent that enables us to do that. And that means that we've been able to enumerate those self-reactive T cells within the repertoire, demonstrating that they're arising because of a defect in the thymus, and then sort those cells and sequence the T cell receptors and demonstrate that sequences within the T cell receptor repertoire, which are normally censored in the thymus, are present in these individuals to drive their autoimmunity. And that's what's summarised in this slide. And then we went back to the patients, the small number of patients, and sequenced their T cell receptors within their polyclonal repertoire, and were able to demonstrate that the same signatures of autoreactivity were present within their repertoire as well. So this means that we've been able to identify the mechanism that's at play within the mouse model and then go back to characterise that in the humans. And this is very important because it means that when we go from monogenic disease to polygenic disease, we've now got a method for doing that. So if we think about a disease like diabetes, type 1 diabetes, which is highly constrained at the MHC level, using this method that we've sorted out in the rare instance of a monogenic disease is now gives us a strategy for approaching early detection in a complex disease. All right, so overall what we're seeking to do in the Grand Challenge is characterise genomic variation, integrate genomics into clinical practice. What I haven't spoken about is too much is how we're going to take information about existing precision therapies to feed into this process. And of course, as I mentioned, there's going to be this comprehensive phenotypic monitoring through wearable devices. So I'm happy to take any questions if there are any, and then I'll hand over to Jane.
Fantastic, thanks, Matt. Great. Questions from the audience or comments? Yeah, that was beautiful work. Fascinating that it was two regulators of transcription, that at least in the examples that you showed. I know you've got a bigger program of research, but...
No, but you raise a really important point because both are actually working in the same pathway, the NF-kappa B pathway, and they're not the only two variants that we've identified in that pathway. So it gives us another, this is another lesson from genomics. Once we start to define a phenotype very carefully, then often once we start to dissect where the defects are, they're at different steps along that pathway.
So you think it's a global loss of self-tolerance at play in these?
Well, in the second example, it's very specific, and I glossed over that, and there are certain organs which are affected, and others which are perfectly normal, and this also is related to the suite of self-antigens which are aberrantly expressed or not expressed within the thymic epithelium.
Yeah. Sorry, I misspoke. It was clearly specific. I was probably more alluding, there's a level of redundancy in these pathways. It is naturally built in, but in this particular scenario, that redundancy has not been able to cope.
Exactly. So this is another really important point, that there's more than one mechanism at play to regulate expression of self-antigen in the thymic epithelium, but there are some weak points.
Yeah, sure. So to perform this sort of work, you need buy-in from siblings and from parents, which I guess is reasonable to obtain. Is it always reasonable to obtain?
It is very interesting that in my experience, and I think the experience of others, that patients with rare diseases are very willing to participate, and families are sometimes strange, but in general, other members are often willing to participate as well if they think that it has the potential to offer some benefit to the affected member of their family. So that's been successful.
Yeah, it's been. I do a little bit of work in rare bleeding disorders, and sometimes it's a challenge, particularly if there's no. I mean, what are we going to do with that information? Clinically.
Yes.
And then, so I was just going to ask around funding and payment. How is this all supported, or is this all sitting in the basic research sphere?
Well, the preliminary work that we've done so far has been funded through through the conventional means, mostly funded through NHMRC. But now as we make this transition to the Grand Challenge, then we have an injection of funds there, which is going to support this, what we hope is the investigation of how we can make this transition to start to apply this more within the clinic.
Yeah, so I mean, in terms of Medicare covered costs, or at least partially covered.
So Medicare is not, none of this is prime time. So even for whole exome sequencing at the moment, we expect that there will be a couple of item numbers coming on for whole exome sequencing relatively soon for rare childhood syndromes, but that will be it. It's going to be some time before whole exome sequencing becomes a rebatable item across the board.
Sure. Bruce?
Thanks, Matt. As you move on to this polygenomic disease model, say with type 1 diabetes or MS, what do you think are going to be the barriers or the hurdles that you have to get over from moving from this method with a single mono?
Yeah, well, I think it's, at the moment, while we can see the pathway for how to proceed, or we can see a couple of pathways, it still takes a lot of time, right? So each of those projects that I've presented in a couple of minutes have run for a number of years, and that remains the case. Even once we're confident that we've identified a gene, and with the advent of CRISPR, which has really expedited the whole discovery process, it's still very time consuming. So at the moment, that's going to be the major obstacle. Well, I don't know if that's quite correct. That is an obstacle. We have, the other major obstacle is the heterogeneity of these diseases. And so if we can start to dissect the heterogeneity, then it might enable us to apply these sorts of techniques more. But again, that just amplifies the size of the task.
Hannah?
I have one here from the app that's been sent through. So it says, what could be the opportunities for early career researchers to ride with these initiatives?
Well, there's lots of opportunities. As I mentioned at the outset, this is a deeply interdisciplinary project, the Grand Challenge, and so a number of colleges and schools across the ANU are participating in this, and we really have opportunities from honours students upwards to be involved in these projects, irrespective of whether they're training in nanoengineering or medicine or biology. So, and I think the other exciting opportunity is that because we do have all of these disciplines talking to one another, there is an opportunity for early career researchers to be doing truly interdisciplinary research. And I think that's really exciting because while we talk about that a lot, there are not too many avenues for early career researchers to really see that in practice.
Okay. Any other comments? No. Thank you very much, Matt.
Okay,
Great
thank you.
talk. Okay. Our next speaker is Dr. Jane Desborough, who is going to continue on this theme, using the ANU Grand Challenge. Jane is a research fellow at the School of Population Health at ANU. She's also a fully registered qualified nurse and midwife, so a fantastic plethora of skills to bring.
Thank you. Great. So actually I'll start off. What I'm going to do is just briefly give an overview and introduce some of the other team members. So as Matt described, our project has three pillars, which is genomics, devices and big data. And then our health experience team is a plank, so we work very closely with all of those pillars. And of course, the whole project is underpinned by two clinical cohorts, one for type 1 diabetes and one for MS. So I'll briefly introduce the leader of the ACT Type 1 Diabetes Cohort is Professor Chris Nolan, and he is very soon to start recruiting young people with type 2 diabetes to the cohort, from very young children through to adolescents and young adults. I think Chris is here to answer questions, or he will arrive, but also Karen Brown, who's one of the team members, is here. So if there are questions later in the session about the cohort, Chris might be here to answer them, or Karen. The person leading the multiple sclerosis cohort is Dr. Anne Bruceley, and she's working very closely with Professor Christian Lueck, Professor Robin Lucas and Dr. Joe Lane. And again, they're not far off, about to recruit to this cohort of people with MS in the ACT and surrounding regional areas. And Professor Lueck is here if anyone has questions after the session. The leader of the devices team is Professor Antonio Tricoli, who many of you would have expected to see here today instead of me, but unfortunately Antonio had another commitment. Antonio, one of his favourite pictures to show is this photo of the world's first magnetic resonance imaging machine in 1977, and now a more recent, immensely improved machine, but still very large and in a building and difficult to access. So the focus of the devices team is looking at the future continuous non-invasive and miniaturised diagnostics, and the development of point-of-care devices for the treatment of MS and type 1 diabetes is one of the foci of our project. Then our data team leader is Associate Professor Hannah Suominen, and Hannah's special area of expertise is machine learning, and of course the role of this team is bringing all of the information together from the sensor devices, from the other data recordings, including genomics, through a bespoke machine learning model to provide customised insight into people's illnesses, regardless of geographical location. So now I'll talk about my area that I know a lot better than the others. And so I'm leading the health experience team. The work our team is doing is underpinned by the concept of patient and public involvement in research. And this refers to research that's conducted with or by members of the public rather than about, for or to them. So involvement refers to members of the public being actively involved with research organisations and in projects, including designing the research, identifying priorities, assisting with data collection, such as interviews, being active co-applicants or grant holders on a project, and being members of an advisory group. Participation, which many of you are familiar with, refers to people taking part in a research study through being recruited to a focus group, a clinical trial, or another type of data collection. And engagement really refers to provision and dissemination of information and knowledge about research through open days or science festivals. Now, I'm going to stop here and tell you a story about how did someone like me end up working on a project with someone like Matt. You can see there's quite a big difference there. So our grand challenge was a challenge put out by the Vice-Chancellor, Professor Brian Smith, to the university for academics from all schools and colleges to come together and collaborate to put together a project that is high risk yet high reward. And so we really did act in the spirit of this, and there was in some of our early meetings groups of us from very diverse areas. And I must say, one of my key memories is sitting in with the group, and I think Matthew was there, and there were physicists, engineers, experts in nanotechnology, and I was thinking, well, I don't actually think I belong here, but I'll stay and hear it out. And we had this expert with nanotechnology telling us how they can devise a device that they sit on someone's eyeball to measure disease biomarkers from the tears. And that they have another device that they can put where the sun doesn't shine. and I was thinking, wow. And then I thought, I put up my hand and I said, I wonder how people feel about having devices put on their eyeball or where the sun doesn't shine. And there was quiet and this moment of reflection. And I think it was in that moment that our health experience team was born. And we are still here. So to start off, before I tell you about how we are working with people living with multiple sclerosis and type 1 diabetes, their families and clinicians on this project, I'm going to play a short video which will introduce some of the members of our team. And I can see it down here. All right, excuse me for a moment. I'm messing things up, aren't I? I just need to get down to that there. Or should I have just pressed the link in the slide?
Maybe just press the link. Go back to presentation. Okay.
Lucky we have plenty of time. I've lost the whole thing, haven't I? Okay.
Oh.
Maybe if we go through into the...
Hang on. Sorry.
No. Maybe if we go actually back into the... Let's go back down to the slide and just press on the link.
Which slide?
I was being very...
down here?
That one, yeah.
Okay.
And if we open it up, and just press there. Okay. And I did have the volume fixed down a bit low. Yes.
When I was first diagnosed with type 1 diabetes, I kind of didn't know what it was. When it's your own child, you're always second guessing whether or not you've got it right, and you're worried that you're going to do something wrong for them.
As health professionals too, I think we put a lot of pressure on the families to achieve good control of diabetes because we want longevity, we want healthy outcomes for our families and their children.
I was diagnosed with MS when I was in my mid-40s, but with the wisdom of hindsight, and when I learned more about the disease, I realised that I was experiencing symptoms, sensory symptoms and visual symptoms in my early 20s.
Not everyone has the same symptoms. It's not a cookie-cutter type disease. Everyone has different symptoms.
Our health in our hands is, I personally think it's a good idea because it gets researchers to hear from people like myself firsthand, and everyone's going to be different in that, but it'll just help them in a way that they can actually see how it affects people and what they can do in different ways.
I think that the major benefit of our health in our hands is actually owning the fact that we have MS.
It does give people, patients, an opportunity to contribute to the body of knowledge about what is actually happening.
We have a wonderful group of scientists who are very keen to get to know these people for whom they're developing technologies and that our research is aiming to help.
There's this one saying that I love. It's just, I don't live with diabetes, diabetes lives with me, so it's basically taking control and doing what I want to do.
Okay, now I just need to get back to... I'll close that one. Okay, so our team, I'm going to give you a brief overview of what our health experience team is looking at, and I won't cover everything, but I'll give you an idea of a few ways that we've started and how we're working with the other teams in the project. So we've got to focus on collaboration and co-design. I'll repeat these questions that are coming up to the right, so I won't go through them now. A focus on data, a focus on technologies, decision-making, and the health services ecosystem. So collaboration and co-design is underpinned by this understanding that each person brings a different lens to anything we're experienced with, but particularly to health. So the way that a doctor or a nurse might look at disease will be very different to the way it's experienced by someone who lives with that disease, by their families and their friends. So our aim for this component was to incorporate patient engagement and leadership and policy engagement at multiple levels in the research from inception to implementation. We started off by forming a health experience advisory board with the aim of having them sit alongside the scientific advisory board to embed this experience throughout the project. We have representatives from advocacy and research organisations on this board, so MS Australia, MS Research Australia, Diabetes Australia, the Juvenile Diabetes Research Foundation, the Healthcare Consumer Association. We have one person with MS and we have one young person with type 1 diabetes, and we also have a lived experience academic from the ANU on this board. The second thing we've done is the aim of this is to build collaborative partnerships with local patient community in each target disease. We've started off by building our research team to incorporate people living with MS and type 1 diabetes. On our MS research team, we have 5 people living with MS, and that includes one partner of one person, and we have a team member with MS who's doing an MPhil, and we also have an MS nurse on our team. In our diabetes team, we have 3 young people with MS. 2 of their parents, we have an accredited diabetes educator, and this is in addition to our academic staff. Of course, our research participants, we will be working very closely with the cohorts to draw on, to get research participants with them. And of course, we're working very closely with ACT Health at the system and the policy and the practice level, and we've been working very closely with them from the beginning of the project. One piece of work we've started on is, they're very foundations we're putting in place here. So it's all well and good to say, we're going to collaborate with people with MS. Well, how do people with MS collaborate with researchers? So we're starting off, we've been funded by MS Research Australia to develop an ideal model of collaboration for people living with MS and researchers. When I refer to people living with MS or type 1 diabetes, this includes people with MS and people who live with them. So it incorporates people with the illness and their families and those close to them. So our aim in this project is to articulate the experiences and priorities of patients as collaborators in research through capturing their experiences in working with researchers and identifying research-worthy problems, identifying differences in the way that people living with MS and scientists conceptualise problems, manage dissenting opinions, and understand the final point of research. And the outcome of this will be the development and implementation of a set of reflective tools to assist this collaboration. And once we have this toolkit, we'd like to test it with our people with diabetes and then see what changes. And we suspect that will be quite a different toolkit and see what changes are required so that for every part of this research, we have something that will assist for us to collaborate in the best possible way. Hannah has given me these dot points, Hannah Suominen, who is here and available to answer questions about machine learning and data. So the 3 aims are to identify disease subtypes and predict disease progression, therapeutic responses, and improve the accuracy and reliability of prognosis, improve disease monitoring and patient outcomes, and to build trustworthiness into this system. So we're starting, our team's working very closely with Hannah to start at point 3, to build trustworthiness into the system using value-sensitive design. So we talk about data and how great it is, Does it enhance understanding, or actually is there just too much of it sometimes? So we asked the question, what are the opportunities, challenges, risks of collecting, integrating and using detailed and specific personalised health data on a large scale? We've started off with a few focus groups with people living with MS, and we've asked them, how do you feel about collecting information about your health and sharing this information? What has very much resonated is it's a lot of work having a chronic illness. It's a lot of work having MS. Everyone experiences this illness quite differently, some less so than others. But what most people said is that by the time they've sort of done the things they have to do about having MS each day, they don't want to think about it anymore. So if collecting information is going to add work and get for them or be an added task, or take their focus back onto how do I feel today, how am I functioning, a lot of them would prefer not to do it. However, many of them at the same time were interested in terms of the big data. Who's going to use this? What are we going to do with it? How will my doctor use this information? A couple of them said, well, people had a very good relationship with their doctor, would say, well, I can plug in a few things that indicate I'm having a bad day. What's he going to do? Ring me up and say, gee, you're having a bad day? And I'd say, yes, I am having a bad day. Now we both know it. So we actually quite enjoyed this reflection of a really lovely relationship with a doctor who obviously is very responsive to this person's needs. That wasn't a common response, and this is actually unpublished work at this point. We're still working with it. But what was universally agreed by people was that if my information is going to help understand this illness and help people down the track, yes, of course, I'm really willing to collect and share my personal health information. And this is our starting point. This small amount of work we've started on at this point. Decision making. This is focusing on the therapeutic relationship. The traditional relationship people have with the medical community is a power relationship, and medical providers have the knowledge and we give it to patients. But things are changing. So how may personalised medicine alter decision making in clinical practice? We have a number of questions to ask about this, but what we do believe is that the more information people have, the greater their expertise is in their health condition. And people, particularly with chronic illnesses, do become experts in their health condition. And that's not always well received. Sometimes it's greatly received. So one of our questions is, how do differences in e-health literacy impact on decision making between clinicians and patients? What happens if you're a clinician and one of your patients comes in with a new device or a new website that they want to use? There's often time constraints, there's knowledge constraints. How is this going to alter decision making in clinical practice? And over time, and as personalised medicine is developed in this project, we're very keen to observe interactions between patients and clinicians around not only continuous monitoring devices, but other forms of personalised medicine. Looking at the devices, so some of the work we've started on is, I'll show you a photo. So here we have our expert patients meeting our expert scientists. One of the questions we're very keen to identify is, what are the ideal device categories and characteristics for use in personal monitoring from a user or patient perspective? So we've started working on, we're nearly finished a systematic review looking at young people's experiences of using devices to manage their type 1 diabetes. And what is very clear from this review is that very few devices have been designed together with young people, informing their design. So here we have a picture of, on the photo in the middle, on the left, is students from the College of Engineering and Computer Studies Engineering Tech Launcher project. So Tech Launcher is an initiative of the College of Engineering and Computer Studies, where they work with clients to develop whatever the client wants them to develop. So this team is working with Professor Antonio Tricoli to develop a breast sensor device, and this was the early prototype. And these are photos of them working with our young people with diabetes, a parent, our diabetes educator, Professor Chris Nolan, and we've really been enjoying that and looking forward to continue this collaboration across the colleges for this part of the project. Looking at the healthcare ecosystem, so we had the wonderful keynote lecture this morning about the GIS work that's being done at the University of Canberra. So we're also interested in how may the health ecosystem change or resist personalised medicine. We do posit that personalised medicine will be a disruptive influence to health services and the health system, and will change the way healthcare is provided and decisions are made. But we're starting off mapping the MS and type 1 diabetes services in the ACT. This map on the right, this is a map of the ACT, but it actually comes from a previous project of Dr. Nasser Bagheri at the Research School of Population Health, and he and Professor Luis Salvador Carula are leading the mapping component of this project. And then we also, it's getting a baseline of what services are in place, but we're doing a qualitative project looking at how do people with MS in the ACT navigate the health system. And that includes the health system in the ACT, but some of them actually go outside of the ACT as well. And also with the understanding that patients get information from all sorts of places now, and we're very interested to know how they seek out information, this peer-to-peer information sharing using social media, what sort of websites they use. So there's 3 components to this, and we've just in the very early days starting the mapping and starting the navigating health care journey maps in the near future. So here's an overview of our team. On the left there are the ANU academics. On the right are our health experience team research collaborators, so people living with MS and type 1 diabetes who are ANU campus visitors. And of course, as with any, this is one team in a very big project, and this is a photo we took at the beginning of our project, just to reflect the diversity of the Our Health in Our Hands team. There are many people not in this photograph, but I think at last count we had about 56 co-investigators, so it's quite a big project. And that's all I have to say. Thank you.
Thank you. Excellent overview. And some of these people are at the John Curtin, but I wasn't aware of this whole arm of the study, so I was aware of Matt's work. And Bruce I know very well, but this is an amazing, sort of value-adding layer. So I imagine, can you comment for a little bit on, you almost alluded to it, the world weariness of the patients, oh, here's another study, here's another questionnaire asking me to fill out. So I imagine you had to get them to sign a consent form, ethics consent form. What was the uptake? What sort of level of uptake do you have? Have you got
for our small project? So, look, it is interesting. I think people with chronic illnesses, their capacity to be involved in research is variable at different times. The local MS community are very keen and very involved in the project. The cohorts will be the main source of research participants, but we've been able to start a little bit of work before the cohorts have started. But we are very aware that on a large project like this, it's very important not to place too much of a burden on participants in the local area, because it is a small area.
So are you capturing the most motivated of the patients?
Well, look, I would imagine that could be the case, and particularly for MS, many people don't disclose to even their families that they have MS. So what we've been fortunate with is earlier in the year we did get quite a bit of media coverage for our project, and we had emails from about 120 people across Australia, very keen to hear more about the project. And some of them in the local and regional areas wanting to be involved. And some of these people are actually disclosing for the first time. So they've been living with MS for a long time and have decided that they'd like to be involved in the project. But yes, I think you're right. I think in any project you do tend to get enthusiastic people on board.
Sure. Other comments from the audience?
Yep.
Just with the diabetes cohort, one of the things that we are quite mindful about is like research burnout. So the main cohort, what the plan for that is that'll involve general, like the annual complication screen. So we want to keep that quite limited, and that'll be like baseline participation. From that, what we plan to do is actually then give the participants or their families opportunity to pick and choose which sub-studies that they take part in. So, you know, they can say yes to as much as they like, no to whatever they want, and, you know, can obviously withdraw at any time from any part of it. But we are actually quite mindful of that. And also through the journey at different times, there's different stresses. So, you know, it will allow them to participate or not.
Drop in and drop out as well, Well, type 1 diabetes, I guess they're mainly paediatric, adolescent
up to young adults. So,
So what is your uptake? How many people on the books?
Oh, we haven't started recruiting to the cohorts. That's in the near future, in the next couple of months, I would imagine.
Bruce? Hang on a sec. Sorry. Make Kath run.
I'm looking at all these faces in front of me here, all these distinguished researchers, and I'm wondering how you have a governance model over this. Have you developed one in terms of how you're going to coordinate and deliver this project?
Look, we have worked very hard on the governance model, and some of the other lead investigators are here, so that you can add to anything I say, Hannah and Christian. There is a governance board at the ANU, and that's with Keith Nugent's office. And then we have our two co-chairs, who are Matthew Cooke and Antonio Tricoli. And we have an executive committee of 7. So the 7, probably the 3 pillars and the 3 planks and one more project CEIs on that. And then we have a larger group of 12, which is sort of 2 from each team. But we have regular meetings, and we are working very closely together in terms of manage the project. We report to the governance board, ANU governance board, twice a year. And at this point, the Health Experience Advisory Board is meeting quarterly, and the Independent Advisory Board, which isn't yet formed, but that will meet twice yearly.
Great, thanks very much, Jane.
Okay, thank you.
Okay, our final speaker is Associate Professor Bruce Shadbolt, who is the Executive Branch Manager of the Centre for Health and Medical Research in the ACT Directorate. And he's going to talk to us on Health Analytics Research Collaborative, or HARC.
Thank you, Liz, for that. I think you just wire me up or something.
You can pop that in your pocket.
Oh, right,
And you can go wherever you want.
Okay, so HARC stands for Health Analytics Research Collaborative, and its aim is to basically collaborate between ACT government and our academics to support high quality research and that's efficient and of high quality, to strengthen strategic partnerships, and to support accelerating the translation of knowledge into practice, policy, and community initiatives. So that sounds pretty impressive. If we can do that, how are we going to do that? I don't know. There's been models in the past, in the last, say, 10 or 15 years here in the ACT, where we've tried to bring in biostatistical support and data experts and so forth, and they've pretty much, just large degree, failed. We haven't been able to really progress very far, and so why have they failed is the question. And a lot of it is because, I think, this is my assessment, is it's been fairly piecemeal. So it's been set up by one university, or it's been in one department or across a particular area, and they've been focused on a particular disease or something like this. And it's really made it difficult to coordinate and be able to develop a whole of system approach. So I suppose this is ACT Health's grand challenge to some degree. I'm with the Centre for Health and Medical Research with the ACT Health Directorate, and I suppose that's where I'm coming from a stewardship role. I'm also with the ANU Biologic Data Science Institute and Medical School here at the ANU. So this is our grand challenge to some degree, and I can see that it would support and help, the previous grand challenge we just heard, in terms of being able to provide them with some of those things they're probably going to labour hard on, where they should have already been provided in some shape or form in reality by an organisation or a service. So if we look at it from a sort of a whole of system approach, I'm going to be covering things such as organisational context, data framework, and an analytics framework. And I can think if one of you are researchers here, or if you're a clinician researcher, you would be interested in a lot of the things we're going to be talking about. And HARC itself we've had a series of workshops last year. It's only been set up for about 9 months now, and they were very well attended across ACT, the territory-wide. We ran 3, and we had over 60 research enabler types come along. In other words, biostatisticians, qualitative researchers, methodologists in other fields, as well as a whole range of different scientists as well, coming to those workshops to try and give us some input as to how we might go about developing and taking this thing through a stepwise process to being a reality. So I'm going to talk about those sort of topics, but in the context of where we've come from a position of collaboration. So this is sort of a model that we've been putting forward. Has come from a very early draft of ACT research strategy. It hasn't been widely circulated yet, but I thought I'd bring it today just to give you an overarching context of where we're looking at it from an organisational point of view. Obviously, we've got it here sitting within a quality and safety framework. We've got this sort of virtual research infrastructure that tries to be strategic, in other words, developing strategically beneficial research, feasible, financially affordable, and performance managed. So we'd like to be able to see research that's within that structure, and that's why I asked that question to Jane earlier on about how is what she's doing governed, because it's really important, as she mentioned, to have that in place. Obviously, a lot of things mentioned about ethics. Ethics is having a sound, or ethics by design, as we like to call it, is critical for being able to, progress with your research, but also being able to make sure that you're doing things in an ethical way, and governance and research support. And there's sort of a whole range of things around IP and fundraising. So most of you would have been confronted those issues at some point in time. It's sort of sitting around clinical services, and we've called it that we're thinking that in here we're looking at things that are co-designed, and we say co-delivered as well, and they're evidence-informed care. So we want to be able to see those types of things, a bit like with the grand challenge we just heard,
from the previous speakers that you have this co-designed, co-delivered approach that's evidence-informed. And sitting around the outside of that, or in terms of if you're thinking about this as territory-wide services or wherever it is, you've got an analytics framework with real-time observational, prediction and prescription types of analyses coming from your data. And coming out of that is, of course, your research outcomes. You need your predictive models, your machine learning, all those other sorts of models that you may want that revolve around patients, caregivers, healthcare providers. And then at the next level, you're looking at healthcare systems and health economic models. And then down to the next level, you're looking at targeted exploratory research, a bit like what we saw with the Hannon Group and their targeted drug discovery and other types of exploratory work that you may do around trying to figure out how we can explore different drug regimes, different models around how we might do precision or personalised medicine. And then you've got your qualitative types of outcomes, which we've heard about as being critical for being able to describe the landscape and being able to help formulate theories and better understanding as you move into sort of the more quantitative models. But that all has to be wrapped around sort of the quality outcomes that you see, or the quality improvement models. So you're trying to look at best patient and caregiver outcomes and experiences. As we just heard from Jane, we're looking at optimised outcome-based care pathways. We're looking at value-based healthcare. If you've been involved with the New South Wales health system, that's how they describe pretty much what they're involved with. Looking at maximising your cost effectiveness of services, your extensive population surveillance and policy, your benchmarking, resilience, satisfied workforce, etc. World-class clinicians, researchers, all coming here. So that's our sort of overarching model we'd like to be able to move towards as sort of a research strategy with some sort of vision. And I think it ticks the boxes for a lot of people who are sitting in here in terms of where they can see they fit in there somewhere. And they're doing something along these lines somewhere. So we think that's probably not a bad model to start with. Again, it is ACT health services focused, of course. It's not in the broader context out in the John Curtin School of Medical Research where you mightn't be engaging with patients at all. It's from our perspective. So then there's sort of got to be some sort of governance structure that's around that. We've got the centre that I'm involved with, which has a stewardship role. So we have a strong interest in priority setting, around governance and around evidence management. So that's sort of a big focus of the centre. In terms of HARC itself, we run the secretariat for HARC. And through this government relationship, we feed into this ACT Health and Wellbeing Partnership Board, which is a partnership collaboration between the universities and ACT Directorate, Canberra Health Services, Calvary, and consumers. And we contribute to this through a research working group. So we have a relationship there. Down here in terms of principles, you can see you've got the research driven by priorities, we've got research driven by high quality and accessible data. So again, this is where HARC plays a role in this. Research driven by well-governed partnerships. Again, we have a role. We're developing or in the process of developing a network of researchers who would belong to HARC, have a membership with HARC. We have research translated into care and prevention. So again, HARC has a role in that in terms of the analytics and being able to translate research into a way to be able to be accessed at the patient care level. And then research that gives a voice to consumers. And that's critical to where we're going, and I'll show you what's happening both at the Commonwealth level and the jurisdiction level to be able to include consumers into all the research, into most of the research that we are involved in. And of course, research has to be ethical by design, and we're looking at developing distributed ethical models to be able to improve that process, make it more efficient and more targeted in terms of the types of ethical approval you're looking for. And that's all then related to your health services and your universities. So in terms of the next step in my content is going down then to the research data governance and management level. And we think that data, and I think everybody would probably agree here, is sort of if you don't have data, if you don't have good data, if you don't have high quality data, if you don't have if you can't access data, it's a big problem. And if you're not used to using data, it can also be a big problem. So we've focused HARC initially in its infancy at being able to develop a data management and governance model for the ACT. And if we look at jurisdictions in the Commonwealth, they've agreed that the following principles underpin health system reform. So the patient is at the centre, research is essential health system activity, research fosters a culture of quality, safety and innovation. You can see access to patients must be easier, partnerships and collaborations, workforce support, knowledge and transparency. So all the things that I've just been talking about are part of what's happening nationally. And if you look around, I was at the National Health Data Summit in Melbourne a couple of months ago, and it's quite amazing what's happening in the other jurisdictions in relation to data and how they analysing data and using data for research and for in creating innovations and quality improvement within the health systems. The examples are numerous, and we've got to catch up and follow these New South Wales and Victoria and Queensland, and they're giving us some great examples of frameworks to follow. So hopefully we'll be able to implement some of those and make our life a bit easier. So in terms of where we're at with the data governance framework, we ran a workshop, a data symposium and workshop in June this year. We had 45 participants. We had organisations represented from the Directorate, from the Canberra Health Services, from the ANU, UC, Illawarra, local health districts, Southern local health districts, Murrumbidgee, ambulance services, New South Wales ACT, PHN, CSRO, ABS and AHW. So we had a really well-represented group of data experts, if you want to call them that, within the audience. So we asked, so the beginning of the symposium was really around talking about data and how we can best utilise data. We had Professor Russell Gruen open with his work that he's doing with the Health Answers Partnership. We had Katie Irvine from Cheryl, which is the Centre for Health Record Linkage from out of New South Wales, which we're a member of, the ACT, and many of you probably use Cheryl, and a number of others. In the afternoon we had the workshop, and we asked these questions. What are your data needs? What principles should be included in your data governance? How do we improve data quality? How do we improve the access to data and collaboration? How do research methods become part of the data governance? And anything else they wanted to address. And it was quite enthusiastically and embraced, and quite a lot of information was provided, and there was quite vocal in terms of from the researcher side to the actual producer's side to the various stakeholders in between. And we've been able to come up with a synthesised set of principles, so they've been extracted from the workshop. I can expand a few on it's a document now. It's a growing document, and I can expand a few on. These are just the high-level principles. I can expand into some of them, but you've got high quality data should be paramount for research. Data are accessible and usable. Data are actively managed and tracked. Consumer collected data accurately reflects the consumer's voice and circumstances, and users of data should be knowledgeable of data and its use. So out of the principle one, we had things like data drawn from sound methodologies that support advancing knowledge, valid, reliable, consistent measurement and visualization tools conforming with international standards, coding systems meeting international requirements for data comparisons with data coded to the highest standard. We had people like NASA from the ANU talking about geocoding and the issues around that. We had others talking about the ICD-10 and the problems around coding. How can we improve those things? So all of these things are becoming part of our principles, which we then will develop a framework to try and take that further within ACT and also New South Wales. They've actually come on board with us so that we've got that ACT and New South Wales surrounding region when we start to look at data. We're doing this, HARC is doing this along with the ACT government to create an infrastructure for projects like the Grand Challenge, your health in your hands. And for others out there, we've tried to create an infrastructure, and your support and your involvement is critical. So it's going to take more than just taking some money here and doing that. It's going to take us all working together. So it's everybody's responsibility, I think, in terms of the data and how we develop high quality data. The other interesting thing was basically the consumer. So co-design research producing data that accurately reflects consumers' experiences and outcomes is a major focus, requiring expertise and training in the tools, methods, and collection of such data. Consumers should be part of the co-design that optimises each consumer's experiences, relying in part on consumer input and feedback on data that reflects experiences and outcomes, and data collected should be culturally safe. So there's a whole range of things that have come out of this workshop that you don't see in other principles and data governance frameworks from, say, New South Wales or even from ACT centrally, that this will add to. So we've added another dimension from a research perspective, which I think is critical as we move forward. If you have a look at some from data that we got from OECD, analyses of how well prepared 10 countries, they only said 10 countries are ready to analyse clinical data for healthcare quality monitoring. So they've just picked on that as a particular focus. But if you look at Australia as a whole, this was in 2015, technical and operational readiness. So technically we're pretty good, where this is high and this is low. And if you look at on this side, which is the data governance readiness, we're pretty dismal. We're pretty much down the bottom. If you look at New Zealand, they're up here, and that reflects in terms of what they're doing in terms of their translation of research into practice. UK, Norway, Ireland, even Poland is on par with us technically and way above us in terms of governance readiness. So I think that's a bit of a telling story. So the effort we're putting into this is worth it. You've got to have these things in place to be able to move forward. And like I said, we've been doing this for 10 or 15 years trying to find a way to move forward, and each of the other processes we've gone through have failed. So I think this is the appropriate way to move. I take it now, so I'm leaving data now and moving on to analytics. And this I've taken from the New South Wales analytics framework. It's quite a nifty looking house almost, I don't know, maybe a house. But they've got here who's going to benefit from the analytics. They've taken a very different approach, New South Wales. I'll have to tell you a little bit about what it means in a second, but I'll just describe this diagram for you first. I'll just keep up with my notes. Okay. So what you've got here is you've got consumers involved, you've got clinicians, you've got the health and other services, you've got health systems, and you've got the population. So they've tried to cover all stakeholders in terms of who's going to benefit from the analytics that they get involved with in terms of their framework. That's pretty critical. Under that, they've got their technology and how they go about developing. A lot of their analytics are machine learning based as opposed to pure statistical predictive types of models. And then under that they have their data. Their driving house is their data. What they've done is they've said, in New South Wales, they've said, we're going to have our research going on over here, and we're really excited about that, and our universities can do a lot of that. And then we're going to have our health services over here. We're going to have an analytics framework that says we want to take your evidence that you've generated from your research, and we want to operationalize that either through an analytical framework or through other means, or through other innovative means. But we're going to take that. We're not going to do further research on it so much, but we're going to operationalize it so that patients can benefit, such as a model for sepsis. They may operationalize a decision tree for sepsis. Or for heparin, they may want to be able to identify who in their hospital is likely to be on heparin and how they can manage that process. So that's their approach at the moment. And we're thinking that's great, and we're thinking that's pretty good, but we want to add to that. We think we want to add that co-design, co-delivery component. So we're looking at being able to develop processes down here that allow us to have an optimization for an individual patient using sort of things like N-of-1 designs. So bringing a research component into these sorts of levels. This part is already known. It's fairly much in the research space. So you've got your qualitative phase, and this is not necessarily linear. I've just drawn that like that. How am I going for time? Okay, I'm nearly finished. Where you can look at the landscape, you can do all those sort of things. You can develop hypotheses, you can look at theory-driven work. On this side, and then you've got your predictive models that you develop through your research, and then from that you're going to be able to take that evidence and put it into practice and try to apply it to an individual patient. So you're moving down to the individuals. And from there, you're going to be able to continue to learn. So we're looking at developing what we call response adaptive designs that use iterative Bayesian statistical learning models within a research design, so an N-of-1 design. And that's sort of the framework in terms of how it looks. I won't go over it too much. It's bringing together the journey, the context, the research technology, and service providers with the consumer in the centre. And we've done most of the mathematical work around this now, so we're pretty confident that that's going to be something we can offer to the health system here. In terms of workforce capability, HARC is part of the Biologic Data Science Institute to a large degree. I have a conjoint appointment with that institute, and we're just hiring. Kathleen O'Brien might be in the audience. We're hiring next week, hopefully. We're interviewing at least an academic level B, and we have an academic level C, I think, who's with us 1 day a week at the moment, they start in the middle of next month, and we've got support staff. So we're starting to build up a bit of a team, and we're also developing a research management system. We've finished off the collaboration network requirements. I won't go further into this, just as telling what our next steps are in terms of the roadmap, where we're trying to operationalise some of these things with that team in terms of some of the algorithms we can develop around the evidence that's already been produced. Target areas where they've got some fairly developed evidence that we can operationalise in terms of making them into algorithms, and work on future projects. Again, a value-based healthcare model. And that's just acknowledging the people that have been involved with us along the way so far, and it's a growing list. And thank you for your attention. I'm sorry I went a little bit over.
Thanks, Bruce. No problem. It's more the live feed. Just worried about losing the live feed. Questions from the audience or comments? Okay. there's one person up the back. Just while the microphone's going, a question from online was, can you explain how this new framework will improve upon the frameworks in existence over the last, 15 years or so, according to the question, in 20 words or less?
Yeah, so they've been fairly piecemeal and pretty much focused on a particular area. So it might be somebody gets a biostatistician that comes over and works in aged care, or they've got a biostatistician that works with the medical school.
Yeah, like a
That's it.
problem.
So it's not fixing the problem. You've got to have a whole of system approach. So we're trying to go from the top down and bottom up, both ways. And that's why I went through those governance structures, the strategic plan, research strategic plan model, and so forth.
So
we think it'll work.
I just wanted to ask what your plan is in terms of getting clinicians involved. Having come through ACT Health as a nursing student, I saw many issues that I didn't see when I did placements in New South Wales and Victoria. And I just wonder how you're going to enable clinicians to take part in your data and your research.
Well, obviously clinicians are very busy, and they obviously don't want to necessarily roll their sleeves up and get involved with this sort of heavy lifting I'm talking about. But once we have that framework in place, they will benefit from that. And they'll also be able to fit into an infrastructure that allows them to relatively easily access data and make the data that they're accessing more efficiently accessed. So then, and also from an importing point of view, when they're actually also data producers, it'll be more efficient as well. So the ACT Directorate just got 70 million to improve the digital solutions here. So it's a real injection from ACT Health in terms of trying to make data more available, easier to import, more efficient in how it's managed. And we've heard from the Director General this morning that research is something that he is extremely focused on and thinks is an extremely important part of ACT and where it's going. So. And
so would HARC cover all research in the ACT or be more ACT Health?
Just ACT Health centric, yes.
Right. All right. Oh, there is real shout.
Really quick one. I just want to add on to that comment if I could, or question. Thank you very much. Kasia Bauer from University of Canberra. I think that when you're talking about co-design, you also want to include clinicians in co-design. While they're the producers and the users of that data, you want to make sure those data elements actually make sense in clinical practice. So I think that there might be elements that need discussion in relation to early involvement rather than later. Can you please keep including that data by the clinicians? They might be busy, but they probably want to be involved earlier rather than later.
Yeah. Definitely. As an example, what's happening in New South Wales is the analytic teams work directly actually at the coalface with the clinicians to develop the whole processes around there, how they develop their analytics. So yes, that's how we'll do it here as well. That's definitely. Usually the voice of the clinician is fairly strong, and we think, you say you're working with a clinician to develop this, but we have to keep reminding ourselves that it's the consumer as well in that model. So I've been a bit silent on the clinician, but yes, definitely the clinician is part of the co-design.
Can I just, Diana Perryman, ACT Health, ANU and UC, when we talk about clinicians, Bruce, I think you might be specifically talking about medical clinicians.
No,
but I want,
not at all.
so I'd just like to point out that there are clinicians, all health professionals are clinicians.
Yes, we have UC represented on our management team through Nick Brown. I'm part of the Allied Health Research Network, and definitely the nursing and allied health and associated students are definitely on our radar.
Great. So I'd like to close the session now and thank all speakers, Bruce, Jane and Matt. Thank everyone for attending.
everybody. As you know, we've got the end of life research day tomorrow. It's going to be busy. Basically, if you're not here pretty early, you probably won't get a seat. However, we do have the ANU auditorium available just over the road in Building 4. So if you haven't managed to get a seat in here, it will be live streamed over in that building as well. And you'll be able to ask questions still of the speakers.